Hello dear students, welcome to your class. Today we are studying NCERT 9th Standard Science Chapter 5, the Fundamental Units of Life Part 2. In the Part 1, we have learned what is a cell, discovery of cells and its components, cell theory, classification of cell based on size, shape and number, structural organization of a cell, cell membrane, cytoplasm, nucleus and types of cells. In the Part 2, we will study structure and functions of cell organelles and cell division.
A cell performs a variety of functions such as production of energy, secretion, etc. These activities of cell are performed by different cell organelles. Students, you know the difference between organ and organelles? Just as there are organs in the body, organelles are inside a cell. Just as each body organ, for example, heart, lungs, etc., performs one or more special functions. Similarly, each cell organelle has a special function in a cell. Today we are going to study about the cell organelles of animal cell and plant cell. Cell organelles are enclosed by membranes. Here inside the structure of the animal cell, here it is the animal cell. Cell organelles are there in the cytoplasm. Here also in the plant cell also. Cell organelles are in the cytoplasm. To understand the function of the cell, it is necessary to know about the structure of cell organelle. Some important cell organelles are Endoplasmic reticulum, Golgi apparatus, Lysosomes, Ribosomes, Mitochondria, Vacuoles and Plastids. Endoplasmic reticulum. It is a large network of membrane-bound tubes and sheets. Here, it is spread throughout the cytoplasm and is continuous with the plasma membrane and nuclear membrane. There are two types of endoplasmic reticulum. Rough endoplasmic reticulum. It looks rough under a microscope because it has ribosomes attached to its surface. Ribosomes synthesize proteins. Next one, smooth endoplasmic reticulum. Its surface is smooth due to the absence of ribosomes. Rough endoplasmic reticulum helps in manufacture of proteins. Smooth endoplasmic reticulum helps in manufacture of lipids or fat molecules. Some of these lipids and these proteins that help in building this cell membrane. Here is the cell membrane, outmost covering of animal cell, and in the case of plant cell, the cell membrane of plant cell also. So that's our lipids and these proteins. Some of the proteins also help in building of this cell membrane of plant cell and animal cell. This process is known as membrane biogenesis. Next, functions. Rough endoplasmic reticulum plays an important role in protein synthesis due to the presence of ribosomes. Endoplasmic reticulum serves as a channel for the transport of materials between here in between the cytoplasm and the nucleus, otherwise in between these various regions of cytoplasm. Here in the cytoplasm, this endoplasmic reticulum that serves as a channel for the transport of materials. Smooth endoplasmic reticulum involved in the synthesis of hormones and lipids. Another function, smooth endoplasmic reticulum plays a crucial role in detoxification of poison and drugs, especially in liver cell of animals. In animals, poisons are absorbed by liver cell. So this smooth endoplasmic reticulum, thus detoxifying many poisons and drugs. Golgi apparatus. Next cell organelle, Golgi apparatus. The Golgi apparatus was first discovered by Camillo Golgi. Scientist name is Camillo Golgi. It consists of membrane-bound fluid-filled vesicles. Here is the membrane-bound fluid-filled vesicles, interconnecting tubules, and smooth, flattened cisternae. Smooth, flattened cisternae. Its functions: Golgi apparatus is involved in the formation of lysosome. Lysosome is another cell organelle. Then it is also responsible for the synthesis of cell wall and cell membrane. Lysosomes, it is a kind of waste dispersal system of the cell. Lysosomes are membrane-bound sacs filled with digestive enzymes. These lysosomes are membrane-bound sacs filled with digestive enzymes. Enzymes are made by rough endoplasmic reticulum. 40 different types of enzymes are there in the lysosomes. 40 different types of enzymes. Lysosomes are also called suicidal bag of the cell because when the cell gets damaged or lysosomes may burst and the enzyme can digest their own cell. So these lysosomes are also called suicidal bag of the cell. Their functions: intracellular digestion. Next function, destroy foreign substances. Foreign substances means any germs or microorganisms. They keep the cell clean by digesting any foreign material that enter the cell. They remove up the bacteria also. They remove up the bacteria. So these lysosomes, otherwise known as suicidal bag of the cell, because when the cell gets damaged, the lysosomes may burst and the enzymes can digest their own cell. Next, that's about ribosomes. Ribosomes that we have already studied. It is made up of ribonucleic acid, RNA and proteins. Main function, protein synthesis. Main function of ribosomes, that's a protein synthesis. So they are called protein factory of the cell. Some of the ribosomes attached to the endoplasmic reticulum. Some other ribosomes that's freely scattered in the cytoplasm. Main function of ribosome, that's the protein synthesis. Mitochondria, rod-shaped structures present in the cytoplasm. Here is the mitochondria. Mitochondria, that's a rod-shaped structures that's seen in the cytoplasm. Each mitochondrion is bound by two membranes. Each mitochondrion is bound by two membrane structure. The outer membrane is very porous, while inner membrane is deeply folded. That's a finger-like projections. These projections are called cristae. And the mitochondria, that's the sites of cellular respiration. So I have already told you that respiration takes place in the cell, in the cytoplasm and mitochondria. Mitochondria are known as the powerhouse of the cell because it generates energy for various chemical activities of the cell in the form of ATP. ATP full form is adenosine triphosphate molecules. This respiration, during respiration, the mitochondria, they generates energy for our chemical activities of our body, and they are stored in the form of ATP. ATP is known as the energy currency of the cell. They have their own DNA and ribosomes. Therefore, mitochondria are able to make their own proteins because main function of ribosome is the protein synthesis. Mitochondria have their own DNA and ribosomes. Then functions of this mitochondria. It releases energy from food in the form of ATP. During respiration, mitochondria release energy from food in the form of ATP. It synthesizes respiratory enzymes also. So mitochondria synthesize respiratory enzymes. Vacuoles are fluid-filled sacs bound by a single membrane called tonoplast. Here is the outer covering of this vacuole. It is called tonoplast. In animal cell, here vacuoles are small sized. Here vacuoles are small sized, but in plant cell vacuoles are large sized. Here is the large sized vacuole. Vacuoles of plants are filled with cell sap. Here these vacuoles that are filled with cell sap containing minerals, amino acids and sugars also. Vacuoles are full of cell sap and provide turgidity and rigidity to the cell in plants. Main functions: give turgidity to the cell because vacuoles are full of cell sap. Many substances like amino acids, sugars, organic acids and proteins are stored in vacuoles. In unicellular organism like amoeba, especially in amoeba, consumed food items are stored in food vacuoles. Next, that's about centrosome. Centrosome is found only in animal cell. It is absent in plant cell. Here is the centrosome. It is located near the nucleus from which spindle fibers develop during cell division. Centrosome is present in animal cell. It is absent in plant cell. It is located near the nucleus. It contains a pair of small hollow granules called centrioles. Main function: initiates and regulates cell division. This is centrioles that initiates and regulates cell division. Plastids. Plastids are found only in plant cell. These are special organelles in different shapes: oval, spherical and disc-shaped. 3 types of plastids: leucoplast, chromoplast and a chloroplast. Here it is the chloroplast. First of all, leucoplast, that's colorless plastids. Depending upon the color, plastids are classified as leucoplast, chromoplast and chloroplast. Leucoplast, these are colorless plastids. They have no pigment. Pigment means coloring substances. They store foods in the form of starch, lipids and proteins. Cells of potato have loads of leucoplast in them. One of the examples of leucoplast is the potato. Next is the chromoplast, that's a colored plastid. These are yellow, orange and reddish in color due to the presence of pigments. Pigments means coloring substances. They are mostly present in petals of flowers and in fruits. Yellow colored pigment. Children, did you see the yellow colored flowers and fruits? Yellow colored pigment, that's coloring substance, is called xanthophyll, which give yellow color to fruits and flowers. Next, orange colored pigment called carotene, which give orange color to carrot. But these violet, blue and purple, these are coloring pigments, but they are not dissolved with the plastids. Instead, they remain dissolved in the cell sap of vacuole, and give that color to the plant structure. Did you see the violet or purple color fruits and flowers? These pigments are called anthocyanin. Next, chloroplast. Chloroplast, that's the green colored plast. This one is the green colored plastid. That's a chloroplast, which possesses the photosynthetic pigment chlorophyll. Chloroplasts are important for photosynthesis in plants. They also have other pigments such as orange and yellow, but these pigments are masked by large quantities of chlorophyll. For example, raw tomatoes and unripe chilies are green due to chlorophyll. During ripening, the chlorophyll degenerates and the masked red, that is carotene, takes over. That's why green color turns into red during ripening. Each chloroplast consists of double membranous envelope. Here, in the structure of chloroplast, this one is the double membranous structure, double membranous envelope. Membrane, its internal organization consists of numerous membrane layers embedded in a material called stroma. Here, inside there are numerous substances. These membrane layers embedded material, that material is called stroma. Inner membrane is arranged along the length of the plastid as lamellae. At certain regions, this lamellae, that's thickened and appear like a pile of coins. Pile of coins means if you are keeping the coins one above the other, likewise. These are called grana. These pile of coins are called, this structure is called grana. Inside this grana, here is the grana. Inside the grana, the chlorophyll is located. Chlorophyll pigment that's located inside the grana. Here inside the grana, this one is the grana. Inside the granum, there are chlorophyll pigments located. Plastids have their own DNA and ribosomes. Functions: leucoplasts are responsible for storing food. Next function, chloroplast imparts various colors to the plant parts. Chloroplast is the site of photosynthesis. Students, explanation about the cell organelles of plant cell and animal cells are over. Hope you understood the differences and similarities of plant cell and animal cell. Here, the parts of animal cell. Here is the parts of plant cell. This is the structure of plant cell. Here is the structure of animal cell. And next, what are the cell organelles? Which parts are the common in animal cell and plant cell? No cell wall in animal cell. Cell wall present in plant cell. Large vacuoles are there in plant cell. Plastids are present in plant cell. Whereas in animal cell, no cell wall, small vacuoles, no plastids. But in both cells, animal cell and plant cell, these are the cell organelles and important parts. Nucleus is present in both cases. Cytoplasm, endoplasmic reticulum, that is smooth endoplasmic reticulum, and rough endoplasmic reticulum is present in both cases. Ribosomes present in both cases. Golgi apparatus and mitochondria are also present in animal cell and plant cell. Our next topic is about cell division. Cell division is one of the most fundamental characteristics of life. It is the process where new cells are produced from the existing ones in order to help in growth, replacement, repair in all living organisms. Cell division necessary for the formation of new cells for growth of the body, then replacement of dead and worn out tissues, and for forming gametes for reproduction. Two types of cell division, mitosis and meiosis. In mitosis, it is the division of cell into two identical cells. Mitosis, it is the division of the cell into two identical daughter cells, with each cell having the same number of chromosomes as in the parent cell. Number of chromosomes as in the parent cell. It occurs in somatic cells. Somatic cells means body cells. Whereas in meiosis, it is the kind of cell division that occurs in the reproductive cells to produce the gametes. The number of chromosomes, here the number of chromosomes is reduced to half. Thus, it is called the reductional division. It occurs in reproductive cells. Meiosis occurs in reproductive cells. The difference between mitosis and meiosis. Mitosis occurs in somatic cells, whereas meiosis occurs in reproductive cells. Here the daughter cells contains the same number of chromosomes that's diploid as that of the parent cell. Two daughter cells are formed. Here two daughter cells are formed. Here in the meiosis, the daughter cells have half the number of chromosomes that's haploid as that of the parent cell, and four daughter cells are formed.
Students, here is the end of the chapter, the fundamental units of life. Hope all of you understood well. Thank you and have a nice day.